Good morning, everyone. This is a great opportunity. Thank you so much for being here this morning. And on behalf of President Sobh and on behalf of the Research Council of the Lawrence Technological University, I'd like to take this opportunity and welcome all of you to our Research Day. This is our 12th research year. Believe it or not, we have been celebrating this for 12 years. And this year actually is very remarkable because the number of posters has gone up significantly compared to last year. So we're so delighted and we're so happy that we have a lot of good undergraduate, outstanding graduate students with their faculty members. So we have, I believe, about 125, Matt? Yes, sir. Wonderful. So please make sure that you take the time, go around, look at them, and talk to the students and see their amazing work with our faculty members. First, I'd like to acknowledge our sponsor. They are here today. World Law Registered Attorney. We have Dr. Bill. He's here. I saw him somewhere. Would you please stand up, please?
Just briefly, we've been partnering now with LTU for several years. Everything from pitch competitions for entrepreneurial events, industry expositions, and well as working with faculty and staff and students in helping protect intellectual property developed here at LTU. We really appreciate our opportunity to be here with you, and thank you very much.
Thanks so much, Bill. I appreciate it. Also,
we will be at the poster session too.
thank you, Bill. Thank you so much. Also, we have the Arbor Hive, and frankly, without their help, we will not be able to print all these posters and will not be able to come up with many things that we'd like to do. So we are very grateful to you, and also for helping our students to check on whatever patents you would like to file. So that would be very good also. And we have been following President Sobh and changing our culture, becoming a research institution. And actually, if you look at our mission now and vision, and if you look at the funding, this year we reach about 9 million plus, and this is great. If you look at the numbers, it looks good. And our mission and our suggestion here that we'd like to be an R2 institution by 2030, and certainly we achieved the first criteria. Now we are moving toward the second criteria, graduating about 20 PhDs, and that's why you see the number of PhD degrees are going up. Today we are celebrating our students, our faculty member scholarship and achievement. And today we are actually celebrating an achievement of two of the great faculty members that we have here today. Shannon and Aleksandra have been selected to be the Presidential Colloquium Awards. Please congratulate them. Please stand up, stand up. Well, Aleksandra and Shannon, this is great because when I read your proposal that you got a very good award for that, I said, wow, this is a role model for all of us, younger faculty member, our students, so certainly they deserve this award, and you will hear from them shortly. Also, for this award, this is a new award. The Research Council, all the deans got together, and they proposed to the president that would like to start a new award. And this new award, as you can see it here, starting this year, and we have faculty members that you're going to see, Sara, Margaret, Jessica, Giscard, Bruce, Areej, Masataka, and my apology if I'm pronouncing your name wrong. But you are going to hear from them in a few minutes. But this is a new award, and we are grateful to President Sobh for agreeing to move forward with this new award to acknowledge our faculty member at different college. Also, we have with us today here, Tina Korani, Professor Korani. Please stand up. Professor Korani came all the way from California, and we are very grateful that she took the time to come all the way to Lawrence Tech and spend a day with us, maybe 2, maybe 3. And we are very grateful. You're going to hear from her shortly. As I said, we have about 125 posters, and Matt has done significant amount of work, and really we are very grateful to Matt for his staying late in the evening. Thank you so much, Matt.
That was easy. You guys do the hard work. Thank you.
Thank you so much, Matt. And we'll be asking our faculty member to be the judging. Please, Matt will be explaining the process later on this morning, and he will be asking you for your help to go around and actually evaluate the posters, and he will be talking about this shortly. As of today, you can see here, our research has gone to about 3.3 million. If you can look at the green number, we are going up. And very soon, that number is going to go much higher. And certainly, we are on our way to become R2 institution, achieving the goal that the president already set for us. If you look for the classification according to this, we became a research university. And you heard about this from the president. He sent a message around. And the next step for us is to be an R2 institution. It's a step in the right direction. And we are very grateful to our faculty member who are doing the great work that will put us on the right map. With this, I'd like to take this moment here and invite our president to come over here and talk to us about his vision for our research. And please join me in welcoming President Tarek Sobh.
Good morning. It is truly a pleasure to welcome all of you to Lawrence Technological University's annual Research Day, a day that celebrates not only the spirit of inquiry, innovation, and discovery, but also you, our distinguished faculty members and scholars and students, the thought leaders who make LTU the powerhouse of intellectual and societal impact that we are. This year's Research Day, as Dean Grace has just mentioned, carries a special sense of momentum. As many of you know, LTU was recently designated as a Carnegie Classified Research University for the first time in our 93-year history. It's a very significant accomplishment, and this national recognition affirms that what we have always known, faculty and staff, that our faculty are doing transformational work that truly matters, work that shapes industries, elevates our profile and various communities around the region and nationally, and propels our students into meaningful careers that makes truly a significant difference. From engineering breakthroughs to entrepreneurial design to cutting-edge advances in health, artificial intelligence, material sciences, architecture, artificial intelligence, and much, much more, your contributions exemplify our motto in action, theory and practice. You're not just producing knowledge, you are applying it, evolving it, and most importantly, sharing it in very impactful ways with the rest of the world. What makes us, Lawrence Technological University, truly distinct is that we do not separate research from education. We embed our research, our projects of significant intellectual merit within our curricula. Our students are not passive observers, they are co-creators, co-authors, innovators, and problem solvers, learning from the best, you, our faculty members. That's because you bring research into the classroom, into the lab, into the community, and into the future. I always say, through well-designed universities, appropriate institutions of higher education are not about dispensing knowledge. They are about creating knowledge and dispensing knowledge. Let me say this very clearly. The work that you do truly matters. It shapes the DNA of this institution. It strengthens our national reputation and ensures that Lawrence Tech remains a destination for those who want to push boundaries, think boldly, and do significant, meaningful works that truly matters, that shapes communities and changes the lives of families, students, and others. Today, as we celebrate your accomplishments and showcase the research that continues to define us, let us also take a moment to envision what's ahead. I think this was laid out very eloquently by Dr. Grace. We are on the path to becoming more research prominent. Whether that means R2, who knows, maybe R1 later. And again, it's all about pushing boundaries. But again, let us also take a moment to envision what this journey would entail. This journey, again, is just the beginning. And with the talent, passion, and brilliance in this room, there is no limit whatsoever to what we can achieve together. I'd like to thank you for your unwavering dedication for research, discovery, but primarily and most importantly to our students. The reason for our existence, for the work that we do in research and teaching and service, is to ensure that our students are well-trained, are able to be capable leaders in the future, and we have been very successful doing this as an institution for the last 93 years. The pace of change is exponential in every discipline of knowledge. The careers of the future are going to be very, very different from what we as faculty and our students have been used to in the past. This work that you're doing, which is of very significant intellectual merit and broader impact, is not an add-on. It's not the icing on the cake. It is crucial for our students' training, for the careers of the future that they will be tackling, and most importantly, for ensuring that they will become lifelong learners who enjoy work of significant scholarship, intellectual merit, and broader impact. Thank you for believing in the vision of Lawrence Tech. Since its founding and what it is becoming and evolving into day after day, I am truly grateful. Here is to another incredible year of innovation, successes, and professional accomplishments. Thank you very much.
Now the moment that we have been waiting for. And I'm going to invite our Presidential Colloquium awardee today to come up here, please, and go on the stage. And I'll tell you, when the Research Council met, no, please go ahead.
Oh, okay.
Please. I'm just trying to. When the Research Council met with all the deans, and the minute the names were proposed, there's no but or if, but we strongly believe that Shannon and Aleksandra deserve this opportunity. Now I'd like to ask Patrick Nelson, Dean Nelson, to join me here to talk and introduce our Presidential Colloquium. Speaker, please.
All right. Good morning, everyone. So, yes, as Dr. Gray said, the deans got together. We looked at faculty across the college for this award. Honestly, this year was easy for me to recommend both Shannon and Aleksandra because of their work on this NIH award. This is the first NIH award that LTU has received where the investigators are solely at LTU. And it's an amazing accomplishment. And it's an accomplishment that is just the beginning of where we're supposed to go. So I don't think there was really any discussion who deserved this. Both of you were very proud of the work that you've done, and I think you'll be very excited to hear the work that they're doing because it's transformative. So with that, it's yours. Thank you.
Thank you. Good morning, everyone. This story begins with chemistry, and specifically a place that many of you maybe have not been before, rural Nova Scotia, where I was born and raised, in a very small community. It's so small that StatsCan doesn't have statistics on population, but it's in the hundreds. You're up on 8 acres of land in the beautiful village of Nova Scotia in New Canada. It's a kind of an odd name, given that it has several hundred people. Never really caught on, but I am from New Canada, Nova Scotia, Canada. And in that community, not many people went to college. My father was a first-generation student from a low-income family, and he always encouraged me to pursue higher education. And he told me that education was the way to a better life. He was one of 7 children, and it was a high school science teacher who encouraged him to go to college. And he earned an undergraduate degree in mathematics with support from my mother and a degree in education. And he spent 30 years as a high school math teacher at our local high school, Bridgewater High School. So all of my high school science teachers were family friends, a very small community where we grew up. And I loved every science class I took. It was a difficult decision, when I went on to college to decide what I was going to major in. I worked hard in school. I wanted to make my father proud, and I ended up graduating high school valedictorian, ranked first in my small class of 70. Our local paper, my mother still has that newspaper article where my valedictorian address was the feature that week. And, and she shared this with me. Even at 17, I told my classmates to be strong, be bold, be courageous, and do not be afraid. Not bad for a naive 17-year-old from rural Nova Scotia. When I went on to college, like I said, loved all my science courses. Originally, I began as a food science and nutrition double major, simply because I had a family friend who I really respected, who was a leader in public health in my community, and I loved, health and medicine. But after my first semester, I realized this degree probably wasn't for me. The content, really didn't reflect what kind of career that I saw for myself. It was one interaction with my freshman chemistry professor who stopped me in the hall after first semester. I earned an A+ in his class, and he said, "Shannon, you're good at chemistry. You should be a chemistry major." And I said, "Sure, sounds good." That was literally the extent of the conversation. I went and changed my major to chemistry and never looked back. Chemistry is a central science that can lead you into so many different fields. After my junior year of college, I saw a flyer in a hallway for an internship in industry, a local company, Biomedica Diagnostics. I was lucky to get that job and work there for 4 months on manufacturing diagnostic materials for testing, specifically for veterinary purposes, and also for water quality testing. I worked with scientists and engineers there to push the boundaries of what was possible in medical science, and I learned a lot from the company founder, Dr. Kiramura, who was involved in the development of the first rapid test for HIV/AIDS and was an immigrant to Canada, originally grew up in Uganda. From the leadership there, that's where I learned about graduate school. As a junior, I hadn't heard of this opportunity before, but Dr. Kiramura and the COO, Dr. Jessi, encouraged me to consider it. And so when I started my senior year in college, I started to do some research on graduate school. So given that I had a love for health and medicine, I decided to apply my chemistry knowledge to study medicinal chemistry. I applied to 2 local graduate schools and recruited my then boyfriend, now husband, to come with me. I said, he was working as a high school teacher at a private high school because neither of us could afford to get an education degree, and we couldn't get student loans either due to our age and financial situation. So I told him, Adam, I heard grad school is free and they'll pay us. Why don't we go get more education this way? And so we applied to the same 2 schools, interviewed at the same 2 schools, got into the same 2 schools, and luckily, we had the same first choice. And so together, we lived in Halifax, Nova Scotia for 5 years, got married after 1 year, and we both finished our PhDs, him in physics and me in chemistry. In my final year of my PhD, there was a seminal article that came out in the esteemed journal Science, and I was amazed. It was an article that used enzymes to make natural product-based drugs. And I thought, I want to learn from this person. That's where I want to do my postdoctoral fellowship. And, you know, my PhD advisor was a little skeptical because I was his first ever PhD student, ever. So our lab was not well established. We weren't a big name that Dr. Thorson would recognize if I sent him a message. But I said, don't worry, I got this. I was accepted to an international conference, the International Carbohydrate Symposium. I was one of the only graduate student presenters. And so I sent Dr. Thorson an email and I invited him to my presentation. He was the keynote speaker at the conference. He came to my presentation, invited me for lunch, flew me to Wisconsin, and offered me the postdoctoral fellowship on the spot. And so it was with that that I learned where Wisconsin was on the map. I was aware it was a US state, but I had never heard of the Midwest. Maybe my second time on a plane, I had no idea where I was going. But I knew where I wanted to work, and I knew I wanted to learn from him. And it was a fantastic experience, you know, working on his research group. We were able to develop a class of drug candidates and, you know, publish a paper and get a patent on those molecules, you know, during my time in Wisconsin. Then my husband happened to be working here at GM at the time. That was his first job after college. And one of his colleagues told him about Lawrence Tech and a job opening here. So I said, well, okay, you know, eventually it's nice to live in the same state as your husband, right? You know, so I'll send him my resume, what's the harm? And so I interviewed here, my first ever interview out of my postdoctoral fellowship, and I got the job. And I've been here for 17 years. You know, I fell in love with teaching. Oh, thank you. I fell in love with teaching and the students and being able to mentor them and help them succeed. So it was an easy decision to stay here. My research broadly focuses in the general area of chemical biology at the interface of, you know, two fields. And I've been involved in a few different research areas, all with the same goal, making molecules that have an impact on human health and the environment. My bread and butter is drug discovery research, and in my lab, we make various analogs and we test them for medicinal activity. I've also been involved in some exciting collaborations relating to osteoarthritis research and degenerative disc disease in collaboration with biomedical engineers at the University of Michigan, University of California, and more recently, the Hospital for Special Surgery in New York currently. I make contrast agents for them that are not commercially available, that allows them to study these conditions and better understand disease progression with the goal of better treatment and diagnosis. And then more recently, I've got involved in toxicology research, and that's the story that Dr. Kuzmanov and I are going to tell you today. So this story of toxicology research focuses on one molecule, BPA. The longer name is bisphenol A, and I expect that many of you will have heard of this molecule before. The ball and stick model is shown here. The black spheres are carbon, the white spheres are hydrogens, and the red spheres are oxygens. This is a molecule used to make a variety of plastics, polymers, that we find in common consumer products shown here. These products are ubiquitous in our environment and in the products that we encounter and use every day. Here's the chemistry for how you might make one of these polymers. Easy, right? No problem. This I teach in my advanced organic chemistry class. And if the students are here, this will be on the final. No? So take note. They can describe to you how to make this polymer. Polycarbonates are commonly found in water bottles, food containers, cash register receipts, and so on. Epoxy resins are the other main class of polymer that is made using bisphenol A and its analogs. And this is the chemical equation, you can say, or scheme used to make those resins. Epoxy resins are common. You might recognize the name epoxy in can linings. in superglue, and in lots of protective coatings. The challenge is that over the past few decades, many researchers have found evidence that bisphenol A is an endocrine disrupting chemical, and it impacts human health and the environment. The endocrine system is kind of like a postal system for hormones. It's distributed, as you can see, throughout your body, and Dr. Kuzmanov can tell you much more than me about how this works. There are many different sources of endocrine disruptors in our environment, in society. Bisphenol A is one major class, as identified by the EPA in this graphic shown here. What was found just over a decade ago was that BPA is particularly impactful for fetuses and young children. And so that was the first push to remove bisphenol A, BPA, from baby products. Canada was the first country in the world to ban the use of bisphenol A in baby products in 2008, and the US followed 4 years later. By that time, most consumers had removed this chemical voluntarily because of consumer pressure. But the question as a chemist should always be, what did they replace it with? Because if you see, the scheme, the chemistry I showed you, you can all imagine, without being chemists, that the chemistry doesn't work if you remove one of the molecules, right? If we could just remove a molecule and get the same sort of physical properties from that polymer, we just wouldn't use it in the first place, because every chemical costs money. So the question was, what are they replacing it with? My oldest son was born at the time when they just removed these products from baby products. And I remember seeing this and not thinking much about it. Great, there's no more harmful chemicals in there. Sounds good as a new parent. And you have many, many things to think about. And then a few years later, I read this popular news article. I like reading about, science in the news. And this headline just really made me feel interested to learn more. That the chemical in these BPA-free water bottles may be just as unhealthy as BPA. And I immediately thought, right, what is in there instead? So, as a scientist, luckily they interviewed the lead PI of the study on which this popular news article was based. And so, when I learned her name, I was able to look up that article and read the PNAS journal article that was published as a result of this popular news article. In the final paragraph of the article, they mentioned that manufacturers had turned to BPS, a structure that I'll show you soon. It's a close chemical cousin of BPA. And they said that BPS was used instead of BPA with little proper toxicology testing to produce BPA products demanded by society. And then the last sentence, they said, there should be a societal push to remove all bisphenols from consumer goods. This piqued my interest because, BPS is just one close chemical cousin. And as a chemist, I wanted to look more into this structure. And I wondered if there was a potential alternative that could still be used to make the plastics that are so prevalent in everyday life, but to make them safer. A very short time later, Dr. Kuzmanov joined our department. And in talking to her, getting to know her, I learned she was a world-leading expert in C. elegans, these tiny worms that she's going to tell you about soon. And I also learned from her, C. elegans, these worms, are what is called a model organism, just like zebrafish, that are commonly used to evaluate molecules to see if they're toxic and study them. So I approached Dr. Kuzmanov, pretty shortly after she came here and talked about this idea, and luckily, she was open to the collaboration. So here's the structure of BPA, if you're wondering. There have been many analogs, close chemical cousins, made that change the groups, the chemical groups, shown in the red circle there, and have produced many different kinds of structures. BPS is shown, and you can see that sulfur and oxygens are there, but the base structure is still prevalent. So for our initial study, I thought, what can we do that's different than what others have done in the literature? So after doing a deep dive to see what had been made before, what kind of alternatives had been tested, I decided that we would try to make some molecules that had a third ring. My theory was that these are there are not many that have been made, and they hadn't been well studied. And I thought, well, this will be structurally different. The 3D structure will be significantly different than BPA. So maybe we should try it and see what happens. See if, adding another ring, very different 3D structure and conformation might cause these molecules to be more healthy, not bind to the same kind of receptors that are causing human health problems. And this was our first article that we published, and we were so proud of this work, which included 6 LTU undergraduate students who worked in our lab tirelessly on this effort over multiple years. We tested BPZ, we made that using a literature protocol, and then we made BPY, and I believe we were the first people in the world to make that molecule, and I got to name it because it looks like BPZ, so I thought, why not BPY? Dr. Kuzmanov will share the results of this testing in just a few minutes. The spoiler alert is, they're not good. So don't get excited yet. After we did this work, we thought, okay, that's not the answer. So much work has been done at that bridging carbon, and just if you do a meta-analysis of the literature, no one was finding exciting results, from making those molecules. And then it was Dr. Kuzmanov that found an article that used this different sort of structure, TMBPF. And she sent it to me to evaluate. And TMBPF, you can see, has a different sort of structure where it's circled in red. It has groups attached to what we call the benzene rings. So it was a significantly different analog that hadn't been well studied before, but there was one journal article that showed that it showed some promise in maybe being different than all the others. So this was the work that we published recently with 3 more undergrad students, and Dr. Kuzmanov will tell you about the results of that testing. Based on those initial preliminary studies, because no one was making TMBPF analogs and testing them, thought this would be a great opportunity to write a grant proposal. We had preliminary results, a promising direction, and so based on this direction, we were awarded on our second submission an R15 grant. Oh, no, thank you. So the first aim, the grant has 2 aims, one chemistry related, one biology related, that you'll hear about soon. The first aim was proposed to make more analogs, make different close chemical cousins of TMBPF. And we're going to test those to try to probe whether some of these different sort of analogs could impact, the way that these molecules bind to receptors that cause human health problems. It's not easy to make these molecules. I'm not going into the details today because it isn't appropriate for this audience, but my students worked tirelessly in the lab to make these molecules. Combining the chemicals, yes, right, Emma, she's cheering and agreeing with me because she's here. The easiest part is combining the chemicals together. The hard part is figuring out what to put in, but even after that, it's easy. The hard part is what comes next, optimizing reaction parameters. So if you don't get what you want the first time, you get side products, you get a low yield. We change a lot of different factors, temperature, time, stoichiometry. We do exhaustive studies to try to improve our reactions and improve the production of our target molecule. We stop the reactions, cool them in the freezer, hope they precipitate, analyze the crude product using a wide variety of analysis techniques to understand what happened. Largely, we usually have to do further purification. We have an amazing automated flash chromatography system we love to use. This is time intensive, but fun and rewarding. And after we think we've purified it, we have to characterize it again using a whole host of different analysis methodologies in order to prove it's the right thing. And this is great. We need to be confident about the molecules we're making before we test them or before they're used by humans. Most of the pharmaceuticals you take are white powders, right? So it's not enough to just make another white powder. We have to figure out what exactly, that white powder is with high confidence. So far, in this year 1 of our grant just concluded, year 1 of 3, and we're making great progress. We've been able to make 3 of the 6 analogs so far in high purity, 2 others impure mixtures that we're working on, developing further, and 1 more that we have to tackle this summer. So I'd like to conclude the chemistry portion of the program now and thank all the undergraduate students who have worked in my lab and done fantastic work on this project. I'm so proud of you and what you've accomplished. I'd like to also thank Dr. Jim Woodworth, our instructional support coordinator, who consistently works hard to keep our instruments up and running, and assists in the lab with whatever we need. And thank my colleagues at Wayne State. We use some mass spec facilities at Wayne State, and the department chair there has been very supportive, in this process and helped us along the way. And of course, our funding sources that Dr. Kuzmanov will tell you more about. So thank you, and please welcome Dr. Kuzmanov.
Thank you. Okay, so you heard the first part of this project about bisphenol synthesis, and I'll tell you more about risk assessment that we do in my lab. But before I start talking about that, a little bit about myself. So I'm originally from Serbia. This is where I went to medical school and earned a medical degree. After graduating, I worked for 3 years as a primary care physician before coming to the US. In here, I attended graduate school at the University of Wyoming. First master's degree in human nutrition, and this is when I worked on studying effect of vitamin D on polycystic ovary syndrome, which is basically a condition that causes hormonal disbalance, irregular menstrual cycles, and affects reproductive health. After that, I did my PhD as well at the University of Wyoming. I switched gears a little bit here and studied genes. This was genetics lab, so studied genes and proteins that are helping C. elegans that you just heard about, the model organism, that are helping them deal with environmental stressors. This was when I was introduced to this wonderful model organism that I still use in my research. After graduating, I moved to California, and as a postdoc, I continued using C. elegans. This time, I studied male fertility, specifically genes involved in meiosis, the process of making reproductive cells. So I studied genes that are important for proper chromosome segregation and basically ensuring a quality. Thank you. Ensuring quality of male reproductive cells. Ooh, loud. Okay, so from California, I moved to Michigan in 2017, joined LTU, and this is when I started my own lab. Since for years I was very interested in reproductive biology, I decided to continue working on this topic. So, as you probably know, in over several decades now, we see a rise in infertility rates. And I bet you know at least someone who's struggling to get pregnant. In fact, 1 in 6 people experience infertility in their lifetime. We know that genetics plays a role, and a certain number of cases could definitely be explained purely on genetics influences. But when we can't explain things with genetics background, we look to environment. You are exposed to, well, all of us are exposed to numerous chemicals in the environment on a daily basis. You're exposed through 3 different routes, actually: ingestion, inhalation, skin contact. And plastics, that we already mentioned, are probably one of the most prominent environmental contaminants. Plastics are everywhere. We use plastic containers to store food, to store beverages. Plastics are equipment, toys, in every aspect of our lives. And not only that, when we dispose of plastics, it will degrade. It will degrade in microparticles, micro and nanoparticles, which again contribute to our exposure. Recent research showed that these particles unfortunately accumulate in our tissues, and they accumulate in almost every tissue in our body, even heart. So knowing all this, now more than ever, we really need to understand what type of impact plastics has on our health. And many research groups are working on this. And as you heard from Dr. Timmons, our focus is bisphenols, which is a plastics additive present in certain types of plastic. BPA, known endocrine disruptor, as you already heard, interferes with hormones in our body. Mainly, it's known for mimicking estrogen, but more and more research shows that this chemical interferes with other hormones in our body. In addition to that, there is more evidence now showing that BPA actually can cause oxidative stress, inflammation, and damage tissues in more ways than just endocrine disruption. Substitutes that you already heard about from Dr. Timmons, BPS, BPF, seem to be not much different from BPA. So there is a high need for appropriate replacement. We're trying that with structural changes, and as we're doing that, we're trying to understand how the structural changes impact its function, and we're looking for the safer alternative, but also trying to understand safe exposure, long-term effects of these chemicals. So, Dr. Timmons is synthesizing chemicals, and in my lab, we are doing risk assessment, testing their toxicity. So let me tell you a little bit about toxicity testing. So for years, mammalian model organisms have been considered as the gold standard in toxicity testing, and that kind of makes sense. If you look at the physiology of mammalian model organisms, they're very similar to us, so we would like to test things in something like mice. However, these model organisms come with certain challenges. First, it's very expensive to house and take care of mice. Experiments take a very long time because their generation time is long, they live for a long time, so sometimes you wait for years to get some results. So because of these limitations, scientists started looking for alternative model organisms, something that's more cost-effective, more efficient, and this is where C. elegans comes in. C. elegans emerged as a wonderful model organism for toxicity testing. First, there are, even though we don't look alike, there are many similarities with humans at the cellular level. So if you look at the biological processes, so that's why we can use them and extrapolate results to humans. Their generation time is short. So they go from an embryo to adult in just 3 days. Also, they live for only 3 weeks. So when you test chemicals, you can get your results much faster, so much more efficient. In addition to that, well, very cheap to keep in the lab, to maintain, and they're transparent, which we love because you could watch cellular processes in vivo without dissecting them under microscope. So this is how we keep them in Petri dishes. If you zoom in a Petri dish, you see? This is how they look like. You see this huge C. elegans on the side. That's actually just 1 millimeter long adult animal that has only around 1,000 cells compared to trillions of cells that humans have. And their generation cycle, as you can see up there, going from an embryo through 4 different larval stages, and then adult takes just 2.5, 3 days. They have 2 sexes. Hermaphrodites on the bottom, which produce both types of reproductive cells, eggs and sperm, and on the top there are males which produce only sperm. So given that they produce both of these types of cells, and the process of meiosis, which is a process of creation reproductive cells, is basically the same as ours, this process is conserved from yeast to humans. We could use them to test how chemicals affect quality of reproductive cells. So back to bisphenols. Bisphenol Y and bisphenol Z were the first bisphenols synthesized, and we used them immediately for testing to assess their toxicity. So in C. elegans, we have a range of tests that we could use to assess toxicity. And I'll tell you a little bit about these tests. These tests have been shown to reliably predict toxicity in humans. So what we do in here, like for example, we look at the brood size. Basically, we expose in animals. Remember, these are hermaphrodites. They produce both sperm and eggs. So you can expose them. They self-fertilize. They have progeny. You can count progeny. Higher toxicity correlates with lower brood size, which makes sense because the chemicals then seems to be affecting number and quality of reproductive cells. So this is one of the assays. Another assay we could use is look at embryonic lethality. So once they lay eggs, you look at hatching. If eggs are not hatching, something went wrong. And usually that's chromosomal abnormalities. So this can point to a very toxic chemical again. Another assay we could use is germ cell apoptosis. Germ cells are reproductive cells. Apoptosis is a programmed cell death. So what happens in these animals when you expose them to a chemical, if a chemical damages reproductive cell will undergo apoptosis. It will kill itself. So basically we could count number of these apoptotic cells. So if you look at this fluorescent image, you'll see the tiny circles. That's how we count. We have a special strain in which we could count as fluorescent circles apoptotic cells. On the side, I'm showing you actually what we see. When the cell is damaged, undergoes apoptosis, basically kills itself, it gets engulfed, it gets eaten by phagocytes, and then on the surface of the membrane that surrounds this apoptotic cell, you have this protein SED1 that we labeled with green fluorescence, and that's how we actually visualize. A little bit more complicated, but just wanted to show you how wonderful, which wonderful things we can use, we can do with C. elegans just because they're transparent, and we could label proteins inside of them with green fluorescent protein and other red and yellow and so on, and we could visualize them under microscope. So we did these assays for BPY, BPZ, and as Dr. Timmons mentioned, they were not much better than BPA and BPS. And as you can see on this graph, with the brood size, I'm just showing you some selected results in here. Brood size was lower following exposure to BPAS, BPY. It was lower in all of them, but statistically significant just in BPY. But when we looked at the number of apoptotic cells, damaged reproductive cells, we saw increase in the number of these cells across bisphenols. So we published this in a time when we published these results. We came across several papers that showed that TMBPF, another bisphenol, seemed to be promising. So first paper suggested this chemical has lower estrogenicity. This means it mimics estrogen to a lower extent. So there is a promise. Probably it won't, the endocrine disrupting potential won't be that high. Then also a C. elegans group tested and compared this chemical to some other bisphenols, and they did it in hermaphrodites, and it seemed that the chemical did not affect reproductive health so much. So we decided to go a step further. We're like, okay, we already know what's going on in hermaphrodites. Let's look into males, into male fertility. TMBPF is not commercially available, so Dr. Timmons synthesized the chemical in her lab, and we tested effect on male fertility. How we do this? Again, you have to use assays that could tell you something about humans, and we know that these specific assays are reliable. So, spermatid morphology. Spermatids are immature reproductive cells. This is the same thing that happens in humans. Reproductive cells are first immature, and then they need to mature and become motile. Remember, like sperm has the flagellum that helps it move, and that's what's important for fertilization. So we first tested effect on spermatid morphology, whether the spermatids have changed shape, diameter, is anything changed there? Then we looked at activation. So as I said, human sperm will move with the flagellum, and in C. elegans they make a little bit different structure called pseudopod, but it's the same idea, it's the movement. So we tested that, then we looked at the brood size. We need to do it a little bit differently in here because we're exposing males now. We have to mate them with females. Again, as I said, we have hermaphrodites in C. elegans, we don't have females, but luckily we can genetically modify C. elegans, and we had the strain which was genetically modified just to produce female reproductive cells, and basically behaves as females. So we have this is the FOG2 mutant in here. So that's how we look at brood size, and eventually we check also embryonic lethality. So results. TMBPF, along with BPA and BPS, affected not the shape of spermatids. Spermatids look fine, but they were a bit smaller. We did not understand why, but we were like, okay, let's check the other things. Even though the spermatids were a little bit smaller, activation was fine following TMBPF. Only BPA affected spermatid activation, and as expected, then the brood size was lower with BPA because, well, if they don't move properly, they won't be able to fertilize an egg. What was the red flag in this is that TMBPF, along other bisphenols, affected embryonic lethality. This suggests that the sperm has some of the chromosomal abnormalities that could affect offspring's well-being. Well, they don't hatch. So this was later, after we published this, a couple other groups showed the same effect in other model organisms, so embryonic lethality, saying that TMBPF basically is not a perfect candidate for replacement. So even though it's not a perfect candidate, it's better than BPA and BPS. So we decided to expand our research, and as Dr. Timmons already explained, synthesize basically analogs of this chemical. This is a scaffold, change it, and we proposed this to NIH, where we got funded. My part is to test it, not only in C. elegans, now using human cell culture as well to test endocrine disrupting ability. We just started this year. Dr. Timmons is synthesizing chemicals in her lab. We're optimizing tests for now to start testing. So far, we tested only one of the TMBPF analogs, A2, in here, using another C. elegans assay, incidence of males. So I'll tell you a little bit how this assay works. So we expose animals to hermaphrodites, to chemicals, and then we count number of males. Appearance of males in C. elegans means meiotic error. Something went wrong in meiosis. So anytime you see a male, that means something went wrong. So why is this? Basically, in C. elegans hermaphrodites, there is 2 X chromosomes. Anytime that they make reproductive cells, eggs and sperm, these 2 X chromosomes need to separate, as it's shown on that image down there. So if they separate properly, all reproductive cells will get X chromosome. When 2 cells that have X chromosomes fuse, you get hermaphrodite. That's 2 X chromosomes. Males in C. elegans are just 1 X. So if something goes wrong, oops, we get a male. Okay? So this is another assay we have, because again, they're transparent, genetically modified. We have a strain in which male embryos glow green. So we could even inside of C. elegans count these male embryos. So this is the assay. We tested several bisphenols, just TMBPF along with A2 analog. It seems for these preliminary results that TMBPF and A2 are looking a little bit better than the rest of the bisphenols. Again, very preliminary. We need to use other assays to continue doing this. But that's so far what we have. Okay, so all these assays that I talked about are great for comparative analysis. Is this bisphenol better than this one? Is this more toxic than the other one? But this doesn't tell us anything about mechanism. What's actually going on? Why these animals are sterile or have impaired fertility? To look into this, you need to look at the molecular level. You need to see which genes are impacted by bisphenols. For this, we actually have collaboration with Wayne State. And recently, we got through CuRE's program, we got another piece of NIH funding for proteomics analysis. So what we're doing in here, we're exposing C. elegans to bisphenols, and we're going to use mass spec to identify and quantify all the proteins in the worm, well, all we can detect with mass spec, right? And then we're going to compare these protein profiles across different bisphenols and among animals that are not exposed to bisphenols. And that will pinpoint to proteins that change following exposure to bisphenols. So that will give us clues what's going on, which processes are affected, which genes, genetic pathways are affected, and tell us more why these animals are experiencing embryonic lethality and problems with the fertility. We're using different strains. I won't talk much about this. You can see hermaphrodites, males, and a couple of different mutants. The idea is all of these are exposed to bisphenols, or all of these will be analyzed with mass spec, so we get these protein profiles, and then we're going to look for overlap, basically to more confidently say these are the key players in these effects of bisphenols. So that's one additional project we're doing. And recently I started collaboration with George Washington University on testing bisphenol impact on immunity. There is a link between BPA and autoimmune diseases, and we want to know whether commonly used bisphenols, as well as these new bisphenols that we're synthesizing, affect immunity. So we're using specifically intracellular pathogen response in C. elegans to test this. This response is very similar to type 1 interferon response in mammals and in humans as well. What we know about this response is if type 1 interferon response is decreased, individuals are more susceptible to infection. This is mostly about viruses when it comes to mammals. If this immune response is overly stimulated, we see sometimes autoimmune diseases, so inappropriate activation of the immune response. How we're doing this, we have a strain in which we could visualize this immune response. So there are proteins that are triggered with this immune response, and so basically, if the immune response is on, we see green animals, right? So we're exposing these animals to, this is just showing when they're infected, because, I'm sorry, I didn't say this, this response is triggered in C. elegans by two shown in here natural pathogens. So what we're doing, we're exposing to bisphenols, and so far, this is very preliminary, we tested BP and BPS. It seems that they're triggering this inappropriately, this response, in the absence of pathogens. So very early, we're gonna be continuing doing this across different bisphenols. So that's a little bit about research that we do in my lab. I would like to thank you for listening. Thank my undergraduate students who so far participated in this project. My collaborators from Wayne State, without them, this proteomic studies, assessment of bisphenol intake in C. elegans would not be possible, as well as my collaborator at George Washington University for the collaboration on the immunity testing. And I would like to thank my department, my college for all the support, and especially Dr. Woodward for being there for any time something's wrong with the equipment in the lab, he has the answers. So thank you, Jim. Thank you so much, and we'd be happy to answer any questions you may have.
Very impressive collaboration between a physician and a chemist. And when they get together, a lot of fancy results, as you can see it. We have a few minutes for a few questions, so please help me out. Questions?
We have microphones up there.
Of course.
In the last example that you were talking about, the BPAs, did they upregulate the immunity?
Yes, they trigger it in the they trigger it in the absence of infection. So we basically just exposed to chemicals. That was the idea, because we know that some chemicals actually can trigger immune response inappropriately when it's not important.
So that's how you get to autoimmune issues.
There is a correlation. For now, we're just doing this assay. It's showing that it's on, but we need really to delve deeper into this to understand. But that's the idea, yeah.
Yeah, that just seems really important for human right now.
Exactly,
we're having so many
and
autoimmune issues.
Exactly. That's why we're using this specific strain, because it was recently shown that this response correlates with the type 1 interferon response, which we know, for example, reduced interferon response correlates with long COVID, for example. So so we're just trying to understand for now very, very early preliminary results.
Thank you. Great talk.
Any other questions?
Any other questions? All right. And by the way, each of you have a poster from your lab on some of this research. So just reminding everybody after Dr. Tina's presentation. Hi, Arun.
Hi, Matt. Hi, Arun D'Souza. I'm visiting today. Fascinating presentations, and congratulations. I was curious about if bio-based alternatives can be used to replace BPA and BBF, or any thoughts on that? I think lignin comes to mind.
Chemistry question. Yes, so there is one paper where researchers have used TMBPF and actually polymerized it and studied the physical and mechanical properties of the polymer. So I think, for the main scaffold that has been created before, I think that was a good citation for the NIH grant and convince reviewers that there is at least, substituting the benzene ring, shows promise in still having the same similar physical properties as without the substitutions. The next stage that I would like to do is, find collaborators, in engineering who could do those sort of tests on the analogs I'm making. I have an industry partner who's interested in making an epoxy using TMBPF. So I was just talking to Mark Brookie about that yesterday. So I'd like to do more collaboration with engineers to test the other analogs. But we do have some evidence, preliminary in the literature, that it could have similar physical properties, because of course, the physical properties, the strength of polycarbonates and epoxies, is extremely important for their ubiquitous use in consumer products.
I hope I asked this question correctly, and then I hope I comprehend the response. So the C. elegans, I'm just wondering about how you, actually I kind of looked it up on my phone quickly too, like how do you, apparently you can grow them, but then how do you ensure like the genetic strain that you're looking for, or how do you ensure that something with the C. elegans is not interfering with your...
No, that's a good question. So all the strains are actually ordered from a specific library where they test mutations. So we know that that specific mutation was introduced when we're using mutant strains. But every time we're testing something, we have a control, a worm that's not exposed to bisphenol. So any change relative to the control would basically suggest this is what the bisphenol is doing. But yeah, that's a very good question. Yeah.
We have a question back here.
One of my questions is, having worked with undergrads, what is some advice that you have for get them engaged, keeping them engaged?
Great question.
I mean, I think it's been impactful to share with students the impact of the project and the reasoning behind it. It's exciting to think that you could be part of science that could be eventually used to improve human health, I think young people are motivated by projects that can improve our world. They're our future. And so that's interesting. I also think having a collaboration has been powerful because they will tell you that they're friends with the biology students down the hall. So to be able to walk down the hall and know that they're producing molecules that are going to be used by their friends to test something and to learn something new is a powerful concept, and I think it's a motivational element for sure when you're not just making the end product to prove that you're a great chemist who can make it in high yield, you're making it for your friends to test, to maybe have a change on society. That's my perception. So Dr. Kuzmanov, you can add.
No, definitely true. Yeah, I think societal impact, as Shannon said, is what they care about, health impact. And again, I noticed that as well. It's really nice, and we're really close. Our labs are really close, and they make chemical and bring in, and then they're asking, oh, did you test it? What's going on? So, yeah.
All right, this is our last question. Okay, we have time for 2 questions. We'll start with Dr. Delago, and then we'll pass it over to Jackie.
Quick question. Beautiful talk and very well-deserved prize. So my question is about So the C. elegans, I'm actually looking now as about 300 neurons, and a Nobel Prize kind of awarded, let's say, animal is the aplysia, has 20,000 neurons, and is really the model for studying memory, right, in neuroscience. So I was just wondering if you know anything about studies about habituation and memory and whatever kind of association done with a C. elegans model.
Great question, and I'll be honest, I don't, but I could connect you with people who do know something about it. So if you're interested in some collaboration with someone, yeah, there is, C. elegans is really great for neurological studies, and I know recently I heard that there are, I think, models for specific human diseases, actually. But yeah, I'm sorry, I'm not an expert on
We need the aplysia for those. All right, so we're going to actually have two more questions, Jackie Moody, and then a student over here.
Hi, my question was, have you guys used the actual C. elegans worms actually from their normal habitat and then compared to those that you have controlled area?
No, so these are lab-grown animals. So there is a strain coming, these are soil worms, actually, and but what we use is actually lab-grown.
Okay.
See, my question is because of as humans are all different. So I was just saying to do the comparison for those actual two different.
Yeah, that's true. But when we're trying to understand what's going on, we don't want many variables in a model organism. So we try to pinpoint what's going on in that animal. But you're absolutely right. After we're done with testing in C. elegans, this has to be confirmed on much more complex animals. Yeah, they are very simple.
Okay, and this is our final question.
So we're from the Lawrence Tech Physician Assistant Program, and it was really interesting that there was like medical-based presentation. So kind of a question we had is, considering the time frame that these species are being exposed to this chemical in comparison to humans, like for example, since the moment we're born, we're kind of exposed to these chemicals. So how can you kind of measure that in a way?
That's a great question, and what we're doing now, that's a collaboration with Wayne State, we're going to determine amount of bisphenols that accumulates in tissues, because that's what matters, right? That concentration needs to mimic what happens in humans. We know specifically for BPS and BPA, we know from other published studies what is the concentration. So we basically have, this is the external concentration we exposed worms to, and then we know that in the tissues, that amount will accumulate for BPA and BPS. We don't know for novel bisphenols, and we are collaborating with Wayne State to determine uptake of the chemical.
Once again, let's give a round of applause to our Presidential Colloquium winners.
I'd like to invite President Sobh and Dr. Kuzmanov and Dr. Timmons to come down here. Dr. Sobh.
Great presentation. Let's have a picture.
Mr. President, if you can stay here. Now, moving forward, I'd like to have Carl. Carl is here today. Yeah. And the College of Architecture and Design, we have Sara and we have Yoshikawa. There, if you can come forward here, please, both of you.
Well, I think it's exciting. We've got we have a team of people from architecture and design, and we see more and more teams as it relates to multidisciplinary work. So the selection of Taka and Sara this year, I think, is both individual and collective. So Taka in the last year has he's won awards from Architects Newspaper. He's presented at the Biennale in Valencia, ACSA National Conference, and the National Conference for Beginning Design Students. Sara then, individually, has done a workshop at UT Austin, had her work exhibited in an invitation for AI, playing models at the UTH in Zurich, Milan Design Week, and was recognized in Italian Design Day. She's also worked to peer review every single class she's taught at LTU. But collectively, in the last year, Taka and Sara have had just simply a graduate research assistant. With the work that they started with that sole graduate research assistant and almost no funding, has presented the work at the ARCC, a national conference, the Society of Ibero-America Geographica Digital, published it in a journal, Officina, and at the California College of Arts Sculpt 2024. That work then has now been included in the Venice Biennale. The Venice Biennale is one of the most prestigious platforms of architectural research and scholarship in the world. It happens every 2 years. The Biennale received thousands of submissions from around the world. Curator Carlo Ratti selected their project for inclusion in the Arsenale in Venice and inclusion in the catalog. Their work is called Fabricated Combines, and it looks at a workflow of AI as it relates to digital design. So I think it's an incredible model for us at LTU, where we don't always have a ton of funding and a ton of support behind what we do, but this group has taken taken their work and taken it internationally into incredible venues. So I wanted to recognize them this year for their work, both individually and collectively.
For the College of Art and Science, I'd like to invite Dean Patrick Nelson to join us here. And again, we have two people that I'd like to invite, Dr. Pell and Jessica. They are here. Would you please come forward? Well,
thank you, boss.
So,
Where are they at? There they come. Hurry up. Can't stand long. Hurry up. All right, so Jessi and Bruce are what I would like to say is two of our stars in our college. And I can say a lot about the work they're doing, but I'm going to change this a little bit than what Carl was doing, because this is the first year that we've done this award. And we've had the research conference now for 12 years. And so we have a lot of faculty here who, if we've been doing this for 12 years, who could be up here getting the same award, because the accomplishments of everyone has been outstanding. And one of the things that happened here when I came here 15 years ago, it's been 15 years already, it's quite surprising it's been that long, and I met with Xiaoping Moore. And Xiaoping had a big transformation on my life in terms of, I was at Michigan, I was doing very well at Michigan, and there was an opportunity, and I wanted to take this opportunity. But what we've done over the last 12 years is significantly change this college. And recently, with people like Bruce and Jessi, is we have faculty who are transforming people's lives. They're transforming what the world is thinking by their research. It's not just publishing a paper to say, oh, I published a paper. That doesn't mean anything. It's what that paper has done to be impactful. Bruce's work on infectious diseases has been outstanding, and obviously I'm a little impartial when it comes to that, because my work is also on infectious disease modeling. but especially to work with COVID. It's really changing the way people are thinking about how things are being treated, and it's making a change in how things are being done. Jessi's work on public policy and social reform is, again, changing people's lives. So the awards for this year was chosen from my perspective on the work that they've done this year. Between the both of them, I think I counted it correctly, 12 publications submitted or published within this last year, many publications in top-tier journals, over $500,000 of funding research brought in. But the big thing here that reminds me is, as we go through this, and I haven't talked to Nabil about this, so this will be a surprise, we have this college award, and I want to sort of name it. And I'm thinking I'm going to propose this to my college because we run a democracy. For when we do the College of Arts and Sciences College Research of the Year Award, I want to name it the Xiaoping Moore Award for research. Because Xiaoping is the one who transformed this, one by bringing me here and getting me to come here and then allowing me to see this, that I do not want to be the best player on the team. I do not want to be the best player or anything. I want everybody to be better than I am. I am a utility player working on a team with a bunch of superstars. And Bruce and Jessi are just a part of these superstars that we have within our college. So thank you.
Well, College of Business. And Areej. Salaymeh, Dr. Salaymeh.
I'm Matthew Cole. I'm the Dean of the College of Business and IT, and I too was hired by Dean Moore. So I really appreciate you doing that, Patrick. You kind of brought tears to my eyes for a second here. And there's a lot of synergy at this campus from a research perspective, and I want to thank Dr. Grace, I want to thank President Sobh, I want to thank the Provost, Richard Heiss, who keeps talking about horizontality and breaking down silos, and Dr. Salaymeh, who I am happy to recognize as the College Researcher of the Year, exemplifies this interdisciplinary work. She has worked really hard this year trying to break into the grant game. I remember one of my first meetings with you when I joined in 2003. We were on the Proposal Transmittal Form Committee. And we also had learn all about at luncheons on how to write grants and get in the grant game. And you said something to me that has resonated all these years. You just got to keep grinding away. You never know when you're going to get funded, but you have to keep trying. And this year, Areej has submitted 5 grant applications, 6, as she's now reminding me, about $2 million total, and they're in her area of machine learning and computer science and information technology, but they are collaborative.
2 of them, I'm the PI of them.
Yeah, 2 of them Areej is the PI on. And the collaboration is with CoAD, the collaboration is with engineering, and collaboration actually with Pedro and Centropolis. We actually just submitted that STTR, so maybe that'll get funded. So again, I just want to acknowledge that this opportunity of the College Researcher of the Year is supported by the Research Council. We pitched it as the deans, as Patrick said, and Dean Daubman and Dean Schutten and Dean Mirmarani. We're very grateful to our faculty who work so hard, and you're not always recognized, right? You get paid, but there's so much more for why you do your work. And we really appreciate it, and this is the day when we get to recognize it. Brings it back to the mission of what this day was about, to recognize research across campus and create a community of research. And I think we have a great community. It's really grown. So, Areej, congratulations.
I'd like to invite the Dean for the College of Engineering to come up here, and Giscard Kfoury, Dr. Kfoury.
Well, good morning. Just want to follow up on what was said before by my colleagues. I'm pretty new here, and when I came here, there are certain things that are obvious. We are not the biggest college of engineering. We are not the richest college of engineering. But what I noticed in my short time that we are probably one of the most talented College of Engineering. There is no shortage of talent. Having been a dean for a long time, I can smell talent from 100 feet away. And boy, the fragrance of talent was in the College of Engineering. So you can imagine.
I should have worked alone. I'm worried about how I smell.
So a quick acknowledgement of my predecessor, Dr. Grace here, who's done a great job hiring really some incredible talented faculty. So you also imagine this must have been a very difficult exercise to just select one person and recognize here. But I had two wonderful associate deans who helped me with this thing. And the nature of this selection goes back to what this person is doing. And that's what we are emphasizing this institution, interdisciplinary. His area of expertise is in controls, and controls is the most interdisciplinary subject in engineering. So, we all hear about automation. Automation plays, and autonomy plays such an important role in everybody's life. We are surrounded by driverless cars, That's part of life in the next 20 years or so. So allow me to just read the bio of Dr. Kfoury, and then we recognize him. Dr. Kfoury received a BS in Mechanical Engineering from the American University of Beirut in 2001, and a master's and a PhD both in Mechanical Engineering from Wayne State University in 2003 and 2008 respectively. His area of expertise are nonlinear control, dynamic systems modeling and simulation, vibration analysis, robotics, and engine dynamics and vibration. He joined Lawrence Tech in 2008 as an assistant professor in the Department of Mechanical, Robotics, and Industrial Engineering when he has served as the director of the Bachelor of Science in Robotics Engineering program from 2011 to 2020 and has been faculty advisor for the Formula, SAE, and the Intelligent Ground Vehicle and Competition teams. His research in the area of robotics, manipulator control, observer design, engine vibration and friction estimation, and experimental engine friction measurement are groundbreaking. His current research interests include hybrid and electric powertrain modeling, control and testing of active suspension systems, medical and rehab robotics, autonomous vehicles, modeling and control, and Industry 4.0 automation. His most noteworthy contribution to the Department, College of Engineering, and LTU is the establishment of the undergraduate robotics lab and securing donation of industrial robot arms and automation equipment to establish a state-of-the-art robotics engineering and automation lab. And that's the Rockwell Industry 4.0 Automation Lab. He also received over $500,000 in funding from Johnson Controls to enhance the Vehicle Engineering Systems Laboratory to enable testing of electrified vehicle and components. During his time at LTU, he submitted over 40 proposals, and his total cash and in-kind dollar grants exceeds $2 million. Through these funded projects, he has hired 25 research assistants and has served as the faculty advisor on numerous master's theses and PhD dissertations, as the faculty advisor or as a committee member. Dr. Kfoury is a member of the American Society of Mechanical Engineers and the Society of Automotive Engineers, and has numerous journal and conference publications, and has served as a reviewer for a number of journals. He also has served as an associate editor for 2 highly popular ASME conferences, including Dynamic Systems and Control and the American Control Conference. As the result of such commendable service, he has been elected to serve in leadership position within the prestigious ASME Dynamic Systems and Control Division. Please join me congratulating Dr. Kfoury.
I watched the money very closely, and no doubt, with all the money that you hear, we are on our way to become an R2.
I have to say this, fragrance of talent. I mean, I'm going to use this, sorry, Marge, I mean, I'm going to use this like probably every day from now on. Sorry.
Margaret?
It's exciting to be at the end of the alphabet as far as the colleges go here. One of the things I want to say, because I want to talk about double digits at some point, so I'm in my 11th month as an employee of LTU. So look at all that veteran time. What I've learned in that time is that my colleagues who have my predecessor, my dean colleagues, and certainly everyone else in this group, and provosts and faculty, you've done a great job of helping collaborate and build the existing programs, particularly in physician's assistant and also in nursing. And those collaborations and that great foundation is really there for us all. And we're really rocking it in the research area. And also, as somebody who's new, I want to tell you, not only are we excited about this notion of interdisciplinarity, I view today as a great way. I've already thought of, with every individual who's talked so far, connections with other people in my unit that would really make us shine and go. So don't be surprised if I'm knocking on your door and saying, hey, have you met this person along the way? So this year, we had a lot to choose from. One of the things that the College of Health Sciences has done in its scholarship definition is it's expanded a bit of the definition from what a typical tech university sees. And that expansion is also not just publications, hardcore research, and the ways that we often do that as well, but also in the scholarship of professional practice. And so our students are working to enhance and expand that profession, and also to be as transforming as everyone else. And so today we're acknowledging one of those people who's making a transformation, and that is Dr. Margaret Glembocki. Sorry about that, Margaret. Yeah. When I first arrived here last summer, she was gone, busy training. She is a trainer for forensic nursing. You can read a lot more about the activity in that research magazine that just came out. There's really fabulous stuff here. And I took it literally, colleagues, that I only had a few minutes, so here I go. We are proud to recognize Dr. Margaret Glembocki, Associate Professor of Nursing and Co-Director of the Nursing Program as the College of Health Sciences Researcher of the Year. Dr. Glembocki is conducting pioneering research at the intersection of social justice, health equity, and trauma-informed care. Her work explores how social determinants and systemic violence influence long-term health outcomes, particularly for individuals impacted by law enforcement-related violence. Through the development of a forensic nursing care model, Dr. Glembocki collaborates with progressive police departments and public agencies to ensure that all individuals in custody, regardless of conviction status, have access to trained forensic nurses. Her scholarship not only elevates the national standard for forensic nursing, but also advances LTU's research mission and exemplifies the transformative power of interdisciplinary innovation. Congratulations.
I'd like to thank all the deans. I'd like to thank everybody here, and congratulations for the Researcher of the Year. And I'd like to thank also the Provost for his hard work. And Richard, we thank you so much for your leadership and for everything you have done to steer this in the right direction. We have about 15 minutes break, and immediately after that, you can see here the clock. So we'll come back here in about 15 minutes, and we're going to have the keynote address. Thank you so much.
Okay. Come on in.
Hello.
Thank you for returning from the break. Before we move on to introducing our keynote speaker, after this session is the poster session. And I really am grateful for all the students and especially the faculty advisors. The way we're doing judging this year, as you walk around the posters, you will see cups on every poster. And you will come visit me if you're interested in judging, and judging is open to faculty and administrators. So please come see me. We've got these fun bag of 700 beads. Now you can take the beads and make a necklace if you'd like, or something else. But what I would appreciate is it. Yes, staff. Apologies, absolutely. Faculty, staff, and administrators. I would like you to, whatever criteria you want to use. Someone gave a great elevator pitch of their poster. Give them a bead. As many beads as you want. If you run out of beads, just come see me. I'll be sitting in the atrium. So without further ado, it is my pleasure to invite Dean Schutten from the College of Health and Sciences to introduce our keynote speaker.
All right. Well, thank you for that welcome. Thank you for that welcome for me. One of the things about being new is that they suddenly say, well, why don't you find the keynote speaker? And then you, as a new person, feel a tremendous pressure to pick from all the many individuals I have interacted with and worked with across time to try to find something that truly would be LTU-ish in its nature. And so I'm very excited today to tell you, first of all, that I spent time as the Dean of the College of Applied Sciences and Arts. Later, that was reimagined as a College of Health and Human Sciences at San Jose State University in the heart of Silicon Valley. And one of the opportunities that I had was to hire a young, you're still young, but yeah, to hire a young assistant professor in the School of Mass Communication whose areas had to do with digital design and lots of things related to media. And across time, I have followed what Tina has done. As I was leaving, I made several connections and inroads to help make San Jose State an Adobe Creative Campus, which is a really fabulous kind of opportunity. And Adobe said, well, who would you recommend that we get as faculty champions? And so I'm bringing you one of those faculty champions today. And she and John are just fabulous advocates for Adobe, and I feel like I should say that San Jose State is really thriving in the digital and AI experience. So let me tell you about Tina Korani. She's a visionary educator, a researcher, and a creative practitioner who serves as an associate professor of digital media at San Jose State. It was a true privilege to hire her and watch her become so dynamic and use her skills to transform across almost every discipline in any university. A cross-disciplinary leader, an Adobe education leader, she's made significant contributions across all colleges at San Jose State, forging collaborations that bridge technology, design, education, and storytelling. As co-founder and faculty member of the Immersive Storytelling Lab in the School of Journalism and Mass Communication, Tina also plays a pivotal role as an Adobe education leader and San Jose State's Creative Campus. Tina champions innovative solutions and applications of augmented reality, virtual reality, and artificial intelligence to advance learning and engage across discipline. Her dedication to inclusive design, human-computer interaction, and digital and creative literacies has not only enriched academic programs university-wide, but has also attracted major external support. Working in partnership with colleagues, and because of her work and her colleagues' work, particularly with the Adobe Creative Campus, it was a big influence that helped San Jose State secure $1 million in funding from Adobe just last year, and $500,000 from Meta to expand immersive media education and applied research initiatives. With a background in user experience and graphic design and an MFA from Louisiana State University, where, by the way, she was presenting on Tuesday. So she's been out and about this week. Tina's work has been recognized with numerous honors, including the California State University Faculty Innovation and Leadership Award, a National Addy Award, which is in the advertising areas, and multiple honors from the American Advertising Federation. Her award-winning projects and widely attended conference presentations highlight the transformative potential of digital storytelling in reshaping education for the future. I was so happy she got to spend the day learning from you all, and she had a great time in doing that, and it's been great. And she's going to talk with us now about how to reimagine the possibilities of creative technology in the classroom and in the laboratory. Thank you, Tina.
Yeah, sorry. Hello, everyone. Again, thank you again for the opportunity to invite me to be a keynote speaker at the LTU Research Day. How are you doing so far? Are you enjoying the program as much as I'm doing? Yeah. Okay. So today, how much time do we have? We have about an hour. Okay. I try to again stay on track, and I have many projects to present, and hopefully. At least one of the projects, the earliest projects you will see is something that you probably saw on your campus. So we're going to see it. Okay. Again, thank you so much, Mary, for the introduction. It's always great, the opportunity you provided, like the connection with Adobe and so many other folks. I think it's been really helping me find the collaborators, find the right connections to grow and just become an Adobe education leader, and then as well as other collaborations. That was, again, thanks to you, Mary. So my name is Tina Korani, and I'm going to talk about a few things today. The first, I'm going to talk about the role of AI in how it can democratize visual communication. And then I'm gonna also talk about how it can customize the learning experience for you. So if you're a visual learner, audio learner, you can just like have AI just customize the educational materials for you. And then I'm going to dive into the immersive storytelling and how immersive environments can help us understand the concept, especially abstract concepts, a little bit easier. And then I'm gonna just end with my where we are going, where we are heading, and just some final takeaways. Oops. Okay. These slides have very cool animation, but that's thanks to Canva and Adobe too. So again, my name is Tina Korani. I'm an associate professor in the School of Journalism and Mass Communication at San Jose State. I mostly teach advertising and public relation courses, as well as some technology courses at San Jose State, including my recent collaboration with HonorsX, their amazing program that I will talk about later about. So I'm also a co-founder of Immersive Storytelling Lab. With my hire in 2017, me and the director of School of Journalism, Dr. Phyllis Johnson, we co-founded the Immersive Storytelling Lab. We're going to learn more about what we do in the lab in the slides. And also, I'm a co-founder of Pionista. It's a fashion brand to empower girls to pursue careers in STEM and leadership. Although my background is visual communication, I'm collaborating with UC San Diego Office of Innovation, one of their faculty science faculty members, who help me find the right market for this and just pitch this idea and everything. And the first line is coming this summer, so I'm gonna share more about my work for Pionista on my LinkedIn. But let's dive into this presentation and learn about the importance of, oops, these slides are moving too fast. The importance of AI in education and visual communication. So let's talk about the moment we're in with the whole AI news. Every single day, we open the internet and there we go, another AI tool, another way of doing things. And sometimes that could be scary for the majority of us, because the skills that we we spend years to sharpen, then it feels like it's useless, because now you can do the same thing with a few clicks. Again, that was my first takeaway of AI. My background being in visual communication, I was feeling that all the skills I acquired in illustration, photography, graphic design, all of that could be is really replaceable. AI can do the whole thing in seconds. So the whole job displacement, all the scary news that came to mind. But then I paused a little bit and I just started to embrace it. Started to think about how anything that for the first time was coming in was scary. The internet, cell phones, this or that. There's always just we still have a lot of people that are still learning some new skills, but AI is just going to help them do things 10 times faster. We still need people to run the machines. So definitely that was the first thing, impression was scared, but then I embraced it. And I had asked my students to embrace it too. So we are just going to learn how AI is breaking down traditional barriers to creativity and innovation. Okay, before I go to the next slide, I just wanted to tell you a little bit about my background. I received my bachelor's in visual communication from Art University of Tehran. Back in time, in Tehran, my studies was more traditional media. So I learned photography, illustration. We had so many different, sketching, drawing courses and everything. But then we had some, again, introduction to Adobe too. But then during my MFA studies at Louisiana State University, that was when I decided to dive deep into technology, not only just Adobe, but also virtual reality, augmented reality a little bit. I explored those a little bit, but also interactive design. I will also show you some of my work from the old times and some of my recent work as well. But then more recently, since I joined San Jose State University in 2017, I mostly explored like visual immersive storytelling and interactive storytelling to communicate ideas, facilitate learning, and all that. So we're going to learn more about those in a little bit. So let's take a look at this.
It doesn't just drive through the future. It moves with it. This is just the beginning.
Okay, why LTU's logo is on there? Anyone has any idea? Okay. Found car concept at LTU. I took a picture yesterday. We were just like exploring some of the maker spaces at LTU, and I found this car. I thought that this is really cool car concept, and it was just like there in one of the labs, and I was like, let me take a picture of this and see like if I have some time tonight to kind of like work on a quick commercial for the car. So let's say it's coming in 2026 with just a few photos on my iPhone, pictures I took on my iPhone. I'm sure that the commercial is probably not 100% there, you know, but probably the designer of the car is gonna upset in this room or maybe. But we only had just a camera and just like I took again 2 photos and then I feed AI the photos, rendered the realistic 3D model, and then use some AI tools to create this commercial in under an hour. So I would say like, hey, we just like taking its time to do the render too on the server. But anyways, it was just like taking something that would usually take about a few days, like from rendering to creating the 3D model or this or that. You can now do it in like hour, in less than an hour. I think that probably in the next few years it's going to be again in seconds. So I thought that it's really cool that this technology can help anyone. Again, no matter if you want to make a commercial, if you want to just like convert your sketches to a final prototype, just take a look at how it's going to look like and then make revisions before we actually go to the building the product. Okay. So what makes an idea stick? I think that was a question that I explored during my graduate studies and then the past couple of years. So we don't just read stories, we see them in our mind. So let's say I was talking about, like, for example, my studies in Art University of Tehran. We kind of like, we kind of understand it, but we don't really can't see it. Like, we need to see visuals to understand them, right? So even sometimes there's no visuals, we try to imagine how this scenario looks like, and that helps us learn the materials better. And that was like one of my challenges back in Iran. I was like probably 8 when I started to realizing that I can not learn some of the materials in my textbooks. It was just like only text. Like back in time, we didn't have we weren't using really the internet. We just had like some textbook with just text. And in our classroom, they were just like asking us just memorize all these pages, and the final is going to be just like if you just even miss a word, you would just like lose a point. It was just like pretty strict, and again, no visuals in the textbook. So I got creative. And I think that's how I started my journey in visual communication. I was actually started drawing the images in the textbook margins to help me remember better. That was like my experience, and I thought that I'm the only one, but apparently there are a lot of people that are like me, are visual learners. We're going to learn more about the different learning modes in the next slides. Okay, so let me tell you a little bit about my work. I think Mary mentioned one of my projects with the blind and visually impaired. Unfortunately, I think I didn't include it here, but that was during my last semester in a grad program. I saw that there is like a school for blind and visually impaired right next to where I was living, and I was like, okay, let me take a look at what their educational materials look like. It was just like, again, curiosity. I already picked my thesis project, and I was like, again, about to graduate in a month. But then I went there again, and then asked the art teacher to help me, just like, let me in their classroom for a day, and just like so that I can just like see how they engage and learn from the textbooks. So when I began going to that classroom, I realized that we as graphic designers are doing, mostly myself, or just critiquing myself as a graphic designer, and not really reaching the audience. We always create for people who have perfect vision. Again, the color accessibility on website and things, those are like, I think it started happening in the recent years, let alone the teaching materials for the blind and visually impaired. So other than Braille, there was like no information on most of the textbooks, no visuals, nothing, and sometimes there were some textures, some of the pages, and that kind of inspired me to work on a poster for the color wheel for blind and visually impaired. The idea was like, I was talking to this student in the art classroom, Claire. She told me that her favorite color, the one that she remembers very vividly, is dark blue. And then that inspired me. I was like, I want to just help her just remember the color a little better. Of course, with my tool set, my limited tool set. So I kind of created a black and white color wheel with primary colors and secondary colors, all with thermal sensors and as well as like audio sensors to just give you the feeling of that color. And again, that was like an experimental project, but that really helped me kind of like empathize with like, with whoever is the user of the product and help them understand the material a little better. So again, I worked in illustration and animation areas. As a graphic designer, I worked for Blend digital marketing agency. And they're in downtown LA, so I work as a UX UI designer, and again, more recently I moved into the film and documentary production. We worked on a documentary about Sacramento Delta and the importance of the water for the California and the nation. So again, more recently, I started collaborating with a lot of science faculties to help them tell their stories in a little more engaging way. So visual learning and AI. So the next few slides, I'm going to show you the importance of visual learning and how AI can really unlock that. To just like the learning, it just can really helps us create visuals. So now that we know it's important to have visuals, like nobody has time to actually start to do drawing on the textbook, and we all have so many other things that we need to do. And so how can AI really help us achieve kind of like the visuals that we need to learn the materials better? So that's like one question that, again, led to this experiment. Do only creative majors use visuals to communicate? Again, we've been all to many of the conferences, and we see most of like the posters, like most of the research presented, they don't accompany visuals, and that's not. Again, most of them are like the students spend years, and professors too, spend years working on their research lab, and they have breakthrough results. Their results are kind of like changing the world, but again, there's only accompanied maybe with a table or like very few visuals. I feel like AI can definitely democratize visual communication and help even science and engineering students create visuals they need to tell their stories better. Doing some research, again, 65% of students are visual learners. And then we process visuals 60,000 times faster than text. And generative AI creates instant visual summaries. Let's just learn about that in the next slide. So it was 2 years ago when we all learned about Midjourney. Midjourney, I think, was one of the earliest generative AI tools that helped us create visuals for materials. So your prompt had to start with imagine. So you just put in imagine and just like put in the prompt, and it would just generate the visual within seconds. So that's why I think we all have to be very imaginative with where AI can take us. Again, mostly I'm talking about generative AI. So this is like a Mona Lisa a portrait, again, one of the experiments with it. Again, for most of these are like experimental projects that I'm presenting here. So again, it took under a few, I think less than 2 hours, that for sure. So I didn't spend too much time, and it was just, again, curiosity is the main reason why I generated them. So again, how can we use AI to bring history and the arts to life? So I feel like now we are at this time and age that we can help all the students, visual learners, learn the materials with just few clicks. So why not doing that? So this is actually, in fact, something that I generated again last night. Again, took seconds to create. You just like feed the machine this image of Abraham Lincoln, and I have a few more. It was hard to choose which one to include, but you can actually have do the lip syncing too. Like platforms like Clink AI, many platforms will enable that. You just again upload an image or a video and ask it to read to you something. So let's say we hear actually Abraham Lincoln's word, not by text, but actually Abraham Lincoln, talking to us, to the students. So again, the visual was created in, this one was ChatGPT, and then then I took that to Sara to animate it. Again, took seconds. Same for this one. I think this is a sculpture. You can just again ask to generate the hyper-realistic model based on the facts. So I never just ask just upload an image and say generate the hyper-realistic version. I just ask AI to do the research, find out about that the story, the age, about that figure, and then ask AI to generate to add the texture, render the hyper-realistic model based on all the facts that we have. Again, for most of these sculptures, we just see those sculptures. We never know how they could look like. This is another one. I think we all know this, Nefertiti. And then you can just again create a video. Again, you can see how scarily realistic some of these visuals look like, and it's been only a year that just you're a year or something around that time that you're able to generate hyper-realistic content based on image references. So just imagine what is going to how the direction we're taking, we're going, how much students can actually learn about the past by helping them understand some of again bringing the history to life and kind of immortalize that. Again, changing leading women in the U.S., historic figures, everyone. You can just now generate hyper-realistic visuals from those old images. You can just also do that with your family photos. You want to just see how your grandparents maybe look like, and you can just again, it will take seconds for the machine to generate the hyper-realistic visuals. And the more information you give AI, the more accurate the result is going to be. It's really important to know how to craft your prompts, what to do before, just generating the visual. It's not always just render this. It's always just giving it the right prompt, the right information, and the accuracy is on point. So how can AI democratize visual communication? Again, there are several tools. One of them is Adobe Firefly. Since I'm an Adobe education leader and then Adobe faculty advisor for our Adobe Ambassador program, I try to always keep a few slides about the Adobe's generative AI tools called Adobe Firefly. It's just one of the really great tools to create hyper-realistic renders again based on the information you have. But again, there are several more. There are probably, I should add 10 more to this list, because probably every week I'm learning about a new tool that is cooler than the other ones. So I just need, if you need any suggestions, let me know. Let's talk about the Adobe Firefly one. So for example, for Adobe Firefly, the things you can do with it is that you can just like feed it some text and it will generate the images for you. But it's generative AI, like the generating images based on text prompt is not the only thing that you can do with it. You can just use the generative fill. If you took a picture like vertical, but now you can just like expand it horizontally. You can use the generative expand. You can just play around with the text effects. So many other cool things. If you have, you already have that feature on some of you on your phone, that if you don't want, someone on that picture, maybe you're not best friends anymore, you can just like take that person off the picture in seconds. But you can also do that in like many of those generative AI tools. But again, you can also create vector graphics. I don't know how many of you are graphic designers here, but you can easily just like generate vector graphics based on your text prompts. And yeah, it's just really cool the how much time some certain things would take. Again, always like I remember the first I was working for a graphic design firm, and they asked me to remove a tree in front of a bank. It was like a very dead, dry tree, and no one wanted to cut it. So they just had me as their intern, like just remove that tree and just spend days on it. And it looks really good, pretty good. Definitely helped me like learn Photoshop better. But then now you can do that in seconds, and why not? Why not, spend more of our time to be creative rather than spending a lot of time, with the things that computers can do. This is like this is 2022. I think that was like the 2022, I think, was at Cornell University. I went to their classroom and I had all the students. Again, this was like when AI just started and everyone was hesitant about it. And then they had me just teach a workshop at their institute to students to help them generate visuals. We just had fun with this one. You can see like the visuals they created is not like really too serious, like an alligator eating pizza for maybe an ad for a pizza place, or a cat by the window, a camel drinking, enjoying the wine in the afternoon. But again, the students were really having fun with this. And that was like when I started to learn, and mostly they were architecture and interior design students. And they just like learned that how much time it could save them, just even editing their own content. So I also use their generative AI a lot in my practice. So they just like give me a few images. I don't know why this one is cropped, but just like they give you a few horizontal images and a few vertical images, and then none of them are framed properly. So you just ask the generative AI to expand the image. Like they just give me like it was cut from here, and then you just like add it. I had to add the rest of it for a newsletter that we were working on, and we definitely wanted the left page to fill up with that image. So again, it can just like really help you with your work. If you're interested, I think this one from a long time ago, but I started posting tutorials on my social media. So if you're interested, just let me know. I can always share the link with you guys later. But it's again Tina Korani if you would like to see what my explorations and tutorials. And then now let's talk about the audio learning at AI. So now we have 65% of students who are learning our materials. We're able to communicate to 65% of our students or our clients. How about the rest of the group? So again, another significant number, 30% of students are audio learners. AI tools transform text into speech, making learning more interactive and accessible. I feel again for audio learners, I personally like not only for audio learners. I'm a visual learner, but the only time that I have to read books is during my commute. So I always like listen to audiobooks. So now you can convert the text to audio. You can convert boring papers. Most papers are not boring, but some of them are a little bit, right? Especially if you're studying 10 papers before your exam. And sometimes we lose students, and AI can definitely help with that. So let's learn how. So this is one of my favorite platforms. It's called Notebook LM. It's like, you just like upload a paper, and it would convert that to a podcast. Again, so you can just like actually upload a couple of papers, like whatever document you have, you just upload the PDFs, And on the right-hand side, it will just summarize, give you a summary of all the materials that you uploaded. You can ask a notebook LM questions about any question. So like, was this mentioned? This name was mentioned in this conversation or any of these documents, and it will tell you. It will give you accurate responses. Unlike ChatGPT, which I'm not talking about the deep research one, but like the general ChatGPT, when you just like ask a question, and every time it could just like the response could be a little bit different. Again, it gets creative with the responses too. Again, most of them are based on facts, but again, it could give you some of the information might not be accurate in generative in like AI tools. But for example, Notebook LM or the deep research by Gemini Pro or even ChatGPT, they actually only give you content from the materials you upload. So this one is one of my favorite ones because I just don't want it to convert it to audio. I also wanted to convert it into a podcast, which is something that I enjoy listening to more than.
Yeah, it's so true. The article, it really dives into how like Shakespeare, he was obsessed with this idea of character. Yeah. He believed the choices that characters make actually like drive the plot forward. So it's not like destiny or some big plan. It's the character's own decisions, like lead them to, triumph or tragedy.
Like think about Macbeth, right? Driven by his ambition, or Othello, he's consumed by jealousy. I mean, they make these choices and it leads them down these like really destructive paths, even though, you can't see glimpses of like their good sides.
Right.
It's that age-old question. Why do good people do bad things?
That's the Areej problem, right? I think we can all relate to that. But
you all definitely want to listen more to see what they're saying and the hosts. But the two hosts are AI. So they are AI hosts. They're actually pretty famous. Like there are a lot of like podcasts that you're listening to. Yesterday I saw they use this two host voices for a baby interviewing a dog. I don't know if you guys, if you listen to that podcast one, it was just like saying like, so what are you? And then the dog was saying, and they had the lip sync too with it, added to it. And it was, oh my God, it was so funny. But you can again do a lot of things with this. Just think about how this can change students' engagement with the educational materials in your classroom. How it can help us understand again some topics which are a little bit not close to our major, right? So, for example, my husband just told me probably 10 times about his thesis, of a PhD thesis. And topic, and then I never understood like what it was talking about, and he just told me like, oh Tina, like I uploaded it in Notebook LM, and now it's like listen to this, and then that was like the time I realized that, oh, now I know what you did. So well done. Okay, so with AI, now that we learned a little bit about the importance of visual learning and audio learning and how AI can just like, again, unlock a lot of, again, possibilities, can help us create visuals, audios, and everything. Well, let's talk about virtual reality. So with virtual reality, you can travel in time, travel in space. These are like something that I generated 3 years ago, these images, 2 years ago or something. But you can just like have a journey to past, to journey to the future, explore the underwater. All of those are like now possible due to, because of virtual reality and augmented reality. So how can XR transform the learning experience? This is a photo of like, I was just like showing an experience in the Children's Discovery Museum in San Jose. And then the experience was mostly, it was just like, multiple groups were just like performing, like it was a 360 video of dance performances. It was mostly for like youth, but I had no clue. A 6-year-old will just like pick up the headset, and he just was in it. He was like immersed, and I was like really worried for the kid. These goggles are like still very heavy, but the kid was just like loving it. He didn't want to come out of it. And then I was like, he also watched it twice. And that was like when I learned that you're talking to a very different generation. They really differently can connect with the visual, with the materials, and can actually learn them in a more engaging way, and why not unlock that? So may I have some water, please? Yeah, it's right there. It's right there. I just forgot to bring it. Thank you. Thank you, Matthew. So okay, so let's learn how VR, AR, and MR can unlock your, it can help people immerse themselves, in the learning environment and learn better. So one of the reasons is that when we are immersed in an environment, we can learn better. So we just put ourselves in someone else's shoes. So when we talk about for example, the refugees camps, we just hear about it, but none of us have been there, seen some things firsthand to really empathize with them. Or there is for training, VR just can help us with no cost, with no risk. Train the workforce to perform better, to just practice something before they actually do a heart surgery, let's say, on someone, right? So VR, it has the power to immerse you in an environment, and to just that will really help you with just the learning and practicing something before you actually go to the field and do that. And again, plus the accessibility. It just makes the materials more accessible to everyone. How many of you guys missed a field trip and then missed the learning that occurred, with that field trip, or due to some accessibility reasons, or the budget, you are not able to go on a trip, and then you miss out. And then VR can actually take the experience to the comfort of your couch and help you still learn and engage the content. So that. Okay, so then we talk about virtual reality. Virtual reality, let me just give you a, just a little summary about what's virtual reality. Virtual reality is those headsets you just put on and then just immerse yourself. It just gives you a full immersion. If I just put on the headset and I can just immerse myself in Florida or Alaska or anywhere else that I want to be, and then it completely disconnects me from where I am right here and puts me in a different place. But augmented reality just overlays content to this area. For example, if you had augmented reality glasses, maybe you could just see some overlays of my maybe next slides, or you could just see my information next to me or something. That's also something that I'm really looking forward to as the technology become more accessible and lightweight. Okay, so some of the labs. So again, as a co-founder of Immersive Storytelling Lab, we get the chance to collaborate with Meta, with many other organizations around us, as well as some of the faculties on campus. So we work with education faculty, create make their materials more accessible and engaging for students, and then we work with the sustainability office, or the environmental science faculty, and help them just create a game and to just help people understand a concept. For example, this choose your own ending game on the right hand side is choose your own adventure type of game. So what you do, which I highly recommend, it was so much fun. You can just we worked together with a few students on that project. So what we did is that, based on the decision you make during the game, there are different chapters. The first one is a fast fashion. Based on the decision you make in that, you're put into different scenarios. So someone asks you to skip the lecture and just go explore the area. And if you say yes, you will go to that area. If you say no, sometimes you don't go there, but again, sometimes you maybe end up the same place. But again, you just decide to buy fast fashion. What are the outcomes? You can actually firsthand see it. And then this will really help us to understand the importance of the decisions that we make. So none of us know what's happening with the fast fashion companies and how they're polluting the environment. So they can actually firsthand see the impact of your actions to the environment. So again, we have many of these projects. Again, as our students are using augmented reality to test different products, and again, they're just exploring it in many different ways. And then we just like create an interface for the product. We just want to test how it looks like. And then this is a student project from 3 years ago. Definitely now it could look hyper realistic in terms of the visuals, and just creating the 3D models. Just like now we can just like ask 3D platforms to kind of build a 3D model for you, and then in seconds you can just probably create the same experience in half the time or less.
In this world, there are many different types of people, and therefore no one looks the same. When it comes to representation and inclusivity, there are groups of people who are left out, and we are often shown what is considered the norm. But who gets to decide what is normal, what is beautiful enough to be on a perfume advertisement? The definition of beauty should not be limited to one skin type when the spectrum of skin is so wide. Inclusivity is important. Walking into an establishment and seeing yourself being represented is empowering, especially when there is a huge lack of representation. Vitiligo is a skin condition that causes loss of skin color.
Okay, so students are like, I'm having students work on it. So I'm teaching in the School of Journalism and Mass Communication. So I have a lot of students who are in PR, advertising, and journalism fields. And some of them were like want to learn the technology, but I always designed the assignment in a way that kind of like make it more educational or create an impact. So now that you have augmented reality, you can just put a QR code somewhere, and you can just put your own ad like in AR somewhere. So for example, I challenge the students to just like look for, just go to, just take a look at the advertisement around them and see if there are anything that could be improved about it. So a lot of students were just talking about the representation and like how most groups don't fit in that box, and then they're not represented in the ad or something. So that was like one the idea behind that AR project. And that AR project, the prototype that was created was again created in under an hour. That was like really, really easy one. Just mostly the idea takes time to come up with an idea, but again, just building it and creating it. We use Adobe Aero for that, which is like really easy, fun tools to use to create AR experiences. But if with that combined with AI, then you can just definitely create an experiences, and you can in under probably 15 minutes. What I also like about AI, augmented reality, is that you can test your products. For example, you design a billboard, you don't know how it's going to look like until you print it and post it on the highway, and like, oh, the font is like not, that easy to read, right? Or like the colors are like doesn't go with the environment. And now with augmented reality, you can just like test it before you actually pay for a penny for printing. So that's actually really helpful. Being a design student, I know how expensive printing could be. And then you just find a typo and then no. I have more student work projects, but again, I don't know if I have time for that. Again, final takeaway, I definitely want to leave some time for questions. Again, education is evolving, and AI and XR are not the future. They're already here. It's just for us, we need to just choose to use it or not use it. I feel like we have to embrace it rather than just like ignore it, because it's not gonna go away. It's just gonna be helping us do things faster. AI just can just like help many people. Like for example, in the these people don't have people to talk to, right? They can still talk to AI and still get that support without, paying someone. Or it's just like, it just helps us in everyday life. It can help us summarize, our findings in a short paragraph. It can help us generate ideas in seconds. It can help us create the product promos in minutes. So why not use this to create, to test, and communicate our ideas? Yeah, so if you have questions, I'm here for the next 10-15 minutes, but you can also reach out to my email, again, tina.korani@sjsu.edu, and then you can also, again, connect on LinkedIn if you have any questions. Yeah. Thank you.
Thank you, Tina. I'm sure we've got a lot of questions. Let's start with Dr. D'Souza. And by the way, Tina will be visiting the poster session for a while before Mary takes her to the airport, so please engage her.
Thank you, Matt. So, Dr. Korani, really enjoyed your presentation, and you had quite the journey. So my question is, let me ruminate for a second. So AI and XR, they collect a lot of personal data from biometric to et cetera, right? And when you talk about the specific intersection of the nexus you just mentioned, AI and XR, I see a big concern from my side because I have background in cybersecurity, and that is because the XR you normally use network platforms, the data address and transmission is very much prone or at risk for cyber attacks. So in your opinion, how does one mitigate it, or what do you think of my question? I guess I'm just
Of course, thank you. Thank you for that question. So I'm not an expert in like security and network security, so I've fed AI some information that I shouldn't have. But that's a very good point. And that's why I think platforms like this AI tools like ChatGPT, they have the .edu, kind of service for some educational institutes. I feel that that's not the only, not only ChatGPT, but also my husband works at Apple, and they have also they were advised to use Gemini Pro, a specific extension of that for their work. So now that all the companies, I think that was like this conversation last year, like we were just getting a lot of emails like about the use of AI. You can't use AI and everything. It's like that was happening across campuses, like everywhere. They were like, we still don't know about it. Again, the security being one thing. The other thing is that we didn't want the students to cheat or like to not put in the work that they needed to learn the materials. But then we all got to the point, like the ChatGPT EDU started at San Jose State, at all the CSUs in April 18, just a couple of weeks ago. Like what did they say? Yeah, like last week. But then, and also my partner, again, who works at the high tech, they were asking them to use the Gemini Pro, a specific version that doesn't share their company's data out to the outside. So that was like, so I think all the institutes are taking a proactive approach before something goes wrong, because a lot of the things that we are feeding AI, we don't know what is going to end up going, right? So AI is just like takes all the information, mix it, and just mash it. But then you might be surprised if you just like take some of your personal data or some of the students' information, or like even takes your design to somewhere else. But so for that, there is a solution, and I think all the institutes most of the institutes are like thinking about like the ways to prevent, the stealing the data, or like just like exposing the information that is not supposed to go outside of the institutes.
Did you have a follow-up? Here you go. Sure.
Sure.
So thank you for that clarity. So it's interesting how I've seen the movie Back to the Future. We've all seen it. From what you're saying, it seems to me that, let me explain. Back in the day when I was much younger, we used to have everything in one data center or but now we have the cloud. Things are everywhere. Things are distributed. You don't know where anything is, right? But from what I interpret from what you said, one of the only ways, apart from better governance, better calibration, model tuning, one way for companies and universities to protect themselves is to take the castle and moat approach, the exact opposite of the anytime, anywhere authorized access, meaning you have models that are on your own data, that are firewalled and segregated and whatnot. Is that essentially what you were saying?
Yes, exactly. I think as long as it all stays in the outside of a company, that they can definitely, the universities have control over that data. Like the institutes, I think, are taking the responsibility, because I mean, how many of you heard about like 23andMe and then the data that was sold? I mean, you sign, I didn't do it, but again, you have contract with these companies, like some of them are like telling you we're not going to use your data, but then until we are get bankrupt and then or just get acquired, and then this other company doesn't have any obligation to not use your data to analyze it or something. So that's why I feel like we should probably trust the more reputable, like again, companies like Google. Hopefully OpenAI. I feel like OpenAI is like becoming like something more. It's not like it's just still. I think it's based in the U.S., right? And so I think it's just still you can just still trust it. And if the university does, like for example with the OpenAI, they already are starting a partnership and just like have the .edu thing with it. So then it looks like it's just still gonna keep the data, everything in the house. So.
Great. So we have another question back here.
Thank you for a great talk.
Of course.
Just my question is, in the future, do you think that because now we are coming to an age in which every AI design and art is actually better than what a human can be. So do you think that in the future we are going to have a market for human imperfection, or it will be that human products are going to be just a hobby?
I think what I didn't discuss today, thank you. What I didn't discuss today was like collaboration. So I feel like being in that area for like, again, exploring it for the past few years, what I noticed is that we're talking about the human AI collaboration. So it's like, it's not, so it's most of our students, even ourselves, we're challenged by teamwork. We always prefer to do things, by ourselves because, we don't have to manage other people. We don't have to, again, project management is another skill, right? Not all of us are excited about that, a teamwork aspect. But I feel like we're going entering a completely different era where we have to collaborate. And instead of like collaborating with a team of 10, we're still gonna collaborate with a team of 10, but like 8 of them being AI tools. So we are, so for example, let's talk about the movie industry. The movie industry, you had to be like how many of you have sent your pitch to Netflix or to, I don't know, DreamWorks? I don't know how many of you are in the films or the animation industry. You never get a response. Like there is no way. So you have no budget. You have very few people who are excited about the film. It's the story, your vision is so clear, and it's the message was worth sharing with the world, right? But now it's kind of like democratized. Like now anyone can create an AI film or animation without just spending. I mean, you still have to pay for some of the subscriptions, but without paying much, like under $100, you can produce a highly hyper-realistic movie. I've seen some of those. They're like, it's very hard to tell if that the actors are not like real people, right? So just like instead of thinking about, it's just taking like the actor's job, it's just like now giving everyone the power to produce a film, to make a film. You just need someone that come up with a story. You can even ask AI to help with the crafting. AI cannot just like craft this story in the most creative manner, right? So you always have to tell what you want, exactly what your concept you have, and it will kind of like could just improve it a little bit for you. But then you have the other person who is studied maybe in the media. So then the two of you together, we want to just work on this short film. I feel like in the next few years, we're going to see a lot of like movies, mind-blowing movies, and they're not put together by Netflix or like this big tech giants. And I feel like that's really empowering because the money doesn't go to the companies. It comes to the people. It comes to the small teams. And some of the teams are just like, again, you'll be surprised as you feel like you're thinking about a group of 10 people. And it could be still a group of 10 people, but the quality of the production looks like 100 people, right? So you don't have to be a tech giant with a lot of money, with a lot of connection to enter some of these film festivals. Just like pair up with one other student, one other person, and work on that film project. After the film is just like an example. Just given any type of project, you don't have to be the giant in that field to be able to do something, because you don't have to pay 10 people's salary to do it. And I feel like we're going to see a lot of startups, so many different startups, that know how to use AI to work to produce films, produce movies, product demos, everything.
That creates opportunities for new businesses, right? That's where we get into the world of entrepreneurship and bringing that curiosity to life. So I think we have time for one more question. Great, here we go. Like I said, though, if you have more questions, Tina's going to be around. So thank you for your last question.
Hi. My question is about authorship and consent when it comes to AI-generated images. There's the story of a woman her headshot was published in an article. She was wearing a different shirt. Seems like it's a small issue, but it was not how she wanted to be represented. And I'm just curious about your thoughts on, there seems to be a fine line, right, when we're talking about like Photoshop editing, using AI to maybe swap out a few things. There's a big gray area, and curious about how you involve that in your teaching as well.
Of course, thank you. So the AI in Photoshop or other tools, it's been there around for like years. I remember when I first started with like Photoshop, I was just like had some pimples and wanted to remove it like very quickly. And of course, and then in Europe, I think a couple of years ago, it was I think against the law to kind of like for the models to do any photo manipulation without just putting it out there, right? So that's also kind of considers like changing your appearance to something else. But I think your question is more like a deeper level, like the ethics of like when AI generates an image, how much responsibility do we have about that image? I feel like right now it's just like the engine that you're using, they're going to be completely responsible. I don't know if it's still in the books. There's a lot of gray areas, as you mentioned. But I feel like we have to put them accountable if they generate an exact copy of somebody else's work or an image or visuals for us. It's not happening anymore. That was like in the past times. I should also admit, for my MFA exhibit in 2017, I produced 20 posters. And I saw like when the first AI hit, that was 2022. I think I started like early 2023. I saw an exact copy of my poster. I got received like millions of like likes and comments and I was like just thinking about like at first I got really like mad about the whole situation, but then I started to think about, okay, they're just using like AI probably. And I mean, it depends on like how you want to take a look at this. I mean, if you want to fight for it, you can definitely do so. Just like say, this is my work and you cannot use it. And definitely they will probably take it down. But I feel like we should just put the platforms, the companies accountable for what they produce. Like if they're getting the, how they're training their models is outside of our, expertise. Like we don't really know how they're training the models. Especially I'm talking about outside of the US, like there's like a lot of countries that also include in their materials, include copyrighted material to train their models, but they don't say it, right? So I think that's like when the question comes like, how can I trust, if what I'm generating is I'm not gonna get flagged, I'm not gonna get flagged or like get any punishment for what's produced. I feel like, again, the companies, first trust the company, like I think some platforms like, I can't say the name of the platforms, but they're not based in US. I think that's for those who should do your research to make sure that they're training non-copyrighted materials, right? But some that they do that, I feel like, hopefully we're human, we're not responsible, we're just using the service and paying for it.
Okay, please give me a round of applause for Tina Korani.